Okay. Hi, I'm Michal Milrad. I'm a scientist here at Elixirgen Scientific, and I'll be moderating the session today. Wanted to welcome you and thank you for joining us at our webinar titled Tools to Study Neurodegeneration, iPSC-derived Neurons, Nerve Organoids, and Engineered Human Synaptogenic Beads. Today, we are excited to have two speakers, Stephanie Napier from Jiksak Bioengineering and Dr. Michaela Schelander, who is a scientist here at Elixirgen Scientific. Before we begin, just go over some of the how the webinar will be structured. Again, we're going to be hearing from two speakers. If you have any questions, you can submit them using the Q&A button at the bottom of your Zoom. And after each speaker, we will have a short Q&A session. If you include the slide number when you write your question, it will allow us and the presenter to refer to the correct slide to better put your question in context. If we do not have the time to address all questions, we will get back to you via email. If your name that you are on Zoom with is the same as the name you used to register, we will be able to find you and get back to you. Our first speaker today is Dr. Michaela Schelander from Elixirgen Scientific. She's a scientist here. Dr. Schelander has a Ph.D. in medical science from the Department of Physiology and Pharmacology at Karolinska Institute in Stockholm. She then came to Baltimore and did a postdoc at the Hussman Institute for Autism before joining Elixirgen Scientific in 2021. Dr. Schelander has over a decade of experience doing research studies with primary cell as well as human pluripotent stem cell-based models. And she particularly specializes in functional disease modeling assays. She is our resident expert in astrocytes and neuronal progenitor cells in particular. And we are excited to hear from her.
Thank you, Michal, for that great introduction. So before diving into the main part of my talk today, I would like to give you an overview of Elixirgen Scientific's operations and technology. So our company was founded in 2016 and is headquartered in the Science and Technology Park at the Johns Hopkins School of Medicine campus in Baltimore, Maryland. We recently also opened a branch in Japan. We have a high level of expertise within our company because more than 2/3 of our R&D department are PhDs. And our core technology, appropriately named quick tissue technology, comprises the use of proprietary transcription factor cocktails to drive fast differentiation of embryonic stem cells or induce pluripotent stem cells into a wide variety of specialized cell types. So here's a general description of how we use our technology to develop cell products and protocols. So first, we identify a short list of transcription factors associated with the desired cell type that we aim to produce. And we use a human gene correlation matrix, as you can see here. And this has been licensed to us by Kyoto University, where it was first developed by our founder and chief scientific officer, Dr. Minoru Ko. And second, we test combinations of these shortlisted transcription factors by means of synthetic mRNA transfection or virus-mediated infection. Third, in combination with an optimized medium formulation, the successful transcription factor cocktail will allow rapid and efficient differentiation of either ESCs or iPSCs to our desired cell type. So this approach has several advantages, and some of them we have highlighted here. And this approach is superior to most other media and growth factor-based protocols, which have varying results. So our company offers a broad range of products and services. So please visit our website to learn more about our kits. Our cryopreserved differentiated cells from healthy donors and disease lines, our neuronal assay ready-to-use plates, as well as our different services, which we're happy to custom make according to your needs. So now to the main part of my talk, in which I will discuss how our quick tissue technology can be used to model human disease in vitro. Through our license with the California Institute of Regenerative Medicine, we have an extensive library of human iPSC lines, both from patients and healthy controls. And we have also successfully collaborated with academic labs to apply our technology to their specific disease line. So some of the models we offer are listed here, and more details can be found on our website. Today, however, I will present data from studies and experiments in which Alzheimer's disease, amyotrophic lateral sclerosis, autism spectrum disorder, and myotonic dystrophy type 1 patient material was used. And through these examples, my aim is to illustrate how our technology and cells can be utilized to generate disease models with relevant phenotypic expression, which subsequently can be used for the discovery and development of pharmacological and gene editing therapeutics. So in this first example, we applied our technology to study Alzheimer's disease, AD. And AD is the most common cause of dementia and abnormal occurrence of amyloid plaques caused by the aggregation of amyloid beta 42 peptides, as well as the accumulation of tau tangles made up by the hyperphosphorylated form of the microtubular protein tau, are still considered some of the main features of the disease and used as biomarkers to detect AD. Here, we produced excitatory neurons using our Sendai virus-mediated quick neuron excitatory differentiating kit. And neurons were derived from a late onset sporadic AD patient carrying the apolipoprotein E, APOE, epsilon 4 variant, which is the strongest genetic factor for Alzheimer's disease. And the patient cells were compared with those from a healthy donor. And then we quantified the amounts of amyloid beta 42 peptides in the supernatants of these cultures, as well as the amount of tau protein in cell lysates using ELISA. And as you can see here, so here on the left-hand side, we have the amyloid beta 42. You can see that it was a progressive accumulation of these peptides, both in healthy and in Alzheimer patient, but it was much more pronounced in the Alzheimer patient. This was also true for the tau protein in the cell lysates. So using our cells, we show that the disease-associated phenotype could be detected and reproduced with these neurons. And the apolipoprotein is furthermore highly expressed in astrocytes. So in this second slide example, we looked for effects of Alzheimer disease in astrocytes. So astrocytes, the apolipoprotein is also crucial for cholesterol processing in the brain carried out by astrocytes, and changes in astrocyte morphology and function are also known to occur in neurodegenerative disorders such as AD. So thus we generated the astrocytes from iPSCs derived from an APOE epsilon 4 carrier. But note in this case, it was a different patient than in the previous experiment where we looked at the excitatory neurons. And we used our quick glia astrocytes and the virus-mediated differentiation kit to generate these astrocytes. And we compared AD patient material with healthy donors. And the astrocytes were cultured for 42 days. And then we analyzed with immunocytochemical detection the expression of GFAP, glial fibrillar acidic protein, which is a common astrocyte marker. Using this marker, we also looked for, we also characterized the three different distinct morphologies of these astrocytes, which also has been published in studies to be altered in Alzheimer's with a preponderance of astrocytes residing in the fibroblast-like state. And so in this study, we indeed found that the 80 astrocytes were residing in the more fibroblast-like state when compared to healthy donors. And we also found that the number of GFAP positive cells were lower in the 80 line compared to the healthy one. So subsequently, the 80 associated phenotype we show here might be used for researching novel biomarkers and drugs designed to impact astrocytes, which has largely been an overlooked target for the treatment of Alzheimer's disease. In our next example, we used our technology to investigate amyotrophic lateral sclerosis, ALS. And so ALS is a fatal neurodegenerative disorder characterized by gradual degeneration and elimination of motor neurons in motor cortex, brainstem, and spinal cord. The TAR DNA binding protein 43, TDP43, is a highly conserved nuclear RNA DNA binding protein involved in the regulation of RNA processing. And the accumulation of TDP43 aggregates in the central nervous system is a common feature of many neurodegenerative diseases such as ALS. So here, iPSCs generated from an ALS patient and a healthy donor were differentiated using our quick neuron cholinergic kit. And the cells were analyzed with ICC at 10 days after differentiation for the presence of TDP43 aggregates. And as can be observed here, pronounced aggregates, as you see here, were found in the ALS patient, which is visualized here in red. So especially in the soma, we found these aggregates. And usually it's supposed to be residing in the nucleus. And this localization pattern of TDP43 was absent in the control line. So this indicates that we can reproduce this disease-associated phenotype with our technology and could be further used to screen for drugs aiming to mitigate the effects of these accumulated proteins. So here is a second study using ALS as a disease model with our technology. So some familiar forms of ALS are caused by genetic mutations in superoxide dismutase 1 gene, but the mechanisms driving the motor neuron disease is unclear. So in the second ALS model, which was a collaboration with the Martin Lab at Johns Hopkins, we assessed the functional phenotype of cells carrying different mutations in this gene. And cells were differentiated using our quick neuron cholinergic kit and co-cultured with human primary astrocytes in a multi-electrode array plate. So using an Axon biosystem master probe, spontaneous electrical activity from neurons sitting on top of the electrodes was recorded. And you can see a representative heat map of the electrical activity when it's recorded by the master probe. And the firing activity readings are then visualized as raster plots. So each of these small spikes here represents a over threshold electrical discharge from the neurons sitting on the electrodes. And from these raster plots, we can then extract information regarding network bursting activity. So the network bursts here are represented or highlighted by the magenta rectangles. So looking at this, we found that the network bursting frequency and the number of network bursts, as well as the amplitudes of network bursts, was diminished in both of the SOD1 mutant lines. So both in the patient line and as well as the gene-edited SOD1 mutated line. when compared to the healthy control line. So this indicates that we indeed can recapitulate an ALS disease phenotype using our patient-derived material with our technology to produce cholinergic neurons. So moving on to our third example, in which we applied our technology to study autism spectrum disorder, ASD. And ASD comprises a group of neurodevelopmental conditions characterized by repetitive behaviors, deficits in sociability, and impaired cognitive flexibility. On a neuronal circuit level, one prevailing hypothesis to the etiology of ASD is the imbalance in excitatory to inhibitory neuron signaling with an increase in excitability. And so here we recorded firing patterns of excitatory neurons using MEA to investigate if the presence of an ASD phenotype could be detected. So using our quick neuron excitatory cell kit, iPSCs from a healthy donor and an ASD patient line were differentiated and cultured with human primary astrocyte for 6 weeks. And as you can see, morphology-wise, we couldn't see a difference really between the ASD and the healthy line. But when we analyzed the firing patterns, we saw that the spontaneous electrical activity, which was recorded for 10 minutes, was altered in the ASD line when compared to healthy control. And so we further quantified this data, and the results show a significant increase in the number of network bursts seen here in the ASD line when compared to the healthy control, as well as a lower number of the average network burst duration time was severely diminished in the ASD line. So both of these metrics indicates hyperexcitability phenotype in the autism line. So using this approach, our cells could be used to provide a screening platform for the development of new drugs aiming to normalize the ASD-associated hyperexcitability phenotype. So in the last example of my talk today, our technology was used to investigate novel treatments for myotonic dystrophy type 1, DM1. And DM1 is a multisystem disorder that affects skeletal and smooth muscle, as well as the eye, heart, the endocrine and central nervous system. And it is caused by a CTG nucleotide repeat expansion within the 3' untranslated region of the dystrophia myotonica protein kinase gene. In this study, a therapeutic CRISPR-Cas9 genome editing approach was investigated in iPSC-derived skeletal muscle fibers from a DM1 patient. And these were produced using our quick muscle skeletal kit. And the disease-specific RNA foci in the DM1 patient line, which can be seen here to the left, was detected using fluorescence in situ hybridization. So you see these small dots, which was made visible by Cy3. And these toxic RNA CUG repeats were successfully eliminated in the gene-corrected line, here to the right, via targeted insertion of polyadenylation signals in the non-coding region upstream of the CTG repeats. So thus, as illustrated in this example, our technology and products can be utilized to develop new and effective therapeutic protocols for the potential treatment of severe diseases such as DM1. So I hope today, through these examples that I presented today, that they have been instructive to show how our suite of differentiated cells and kits can be used in the modeling of human diseases and subsequently might aid the investigation and discovery of new therapeutic protocols and pharmacological inventions. So please visit our website or contact us directly to discover how our technology could help you to advance your research and attain your scientific goals. So with that, I thank you all for your kind attention, and now I will take your questions.
Thank you so much, Dr. Schlander. So we do have some questions for you. On slide 12, what percent of the TDP-43 cells had those cytoplasmic aggregates?
So I'm not aware that we did any quantification of this. So I would have to go back and look at our data to be fully confident in giving any kind of answer to that question. So I apologize for not knowing the answer, but I will take a look at it.
Yeah, I'm not sure about this particular study, but I know that for other clients in similar studies, we have done quantification. So.
Sure.
Have you studied the effects of the AD astrocytes on neurons?
So we have not done this yet, but that's a very interesting proposal. And I know in others in the literature, it has been reported that mixing AD line astrocytes with healthy neurons could help us to really investigate how the effect of astrocytes are on neurons. And since these are overlooked cell type in the disease, it would be highly desirable to know this more in detail.
Okay, on slide 14, what symptoms of ASD do you interpret as abnormalities in the network burst of iPS neurons derived from the ASD patients?
It's a very good question. So when, in this case, we compared it to the healthy donor, and when the network bursts are so frequent and it could really be classified as a hyperexcitability, kind of epileptic, seizure-like state of the cells. So I'm not sure if that's answering the question in detail, but I would love to discuss this further as well. And
okay, thanks. I think that's actually all the time we have right now for questions. We have another talk coming up from Stephanie. So Stephanie Napier is joining us today from Jiksak Bioengineering, where she is the manager of the global business development. She has a bachelor's in science in biology from the University of South Carolina, and then went on to conduct research at the Center for Cell and Gene Therapy at Bayer College of Medicine at the University of Tokyo. Stephanie also managed the flow cytometry unit at Quintiles, Japan, before joining in 2016 TSERA, which is a partnership between Takeda and Kyoto University. And there she gained a lot of experience with iPSC. In 2021, Stephanie joined Jiksak first as a scientist, and now she is in a business development role. Again, she's the global business development manager. So thank you for joining us, Stephanie.
Thank you for that great introduction, Michal, and thank you to Elixirgen Scientific for allowing us to present our technology today in this webinar. Are you able to see my screen? Okay, so yeah, As Nihal said, my name is Stephanie Napier, and I am the manager of global business development at Jiksak Bioengineering. We are located right outside of Tokyo in Kawasaki, Japan. So it's 11 o'clock here at night. So our mission at Jiksak is to make ALS a treatable disease. And I know in the last presentation, there was a bit of introduction about ALS. I'm sure that many of the participants today are familiar with ALS, but I just want to highlight that amyotrophic lateral sclerosis, or ALS, is a devastating disease that is characterized by progressive muscle weakness that inevitably culminates in respiratory failure and death within usually 2 to 5 years after diagnosis. At the current time, there's no cure, and there are very few treatments for ALS. And it is our hope that our technology will lead to treatments for this devastating and fatal condition. Now, I'd like to go into some of the challenges that we face in neurological research and talk about how perhaps our technology can help with those. So, for many diseases, we do not fully understand the mechanisms behind what causes the diseases. Not only that, but while we may know the genes that cause some of the diseases, there are many aspects there are some strains of diseases that we do not know the genes for at all. So using our technology, we believe that we can help elucidate disease mechanisms, and through using genetically manipulated material, we can find the genes behind some of these conditions. Not only that, but when it comes to neurological conditions particularly, there are a real lack of relevant animal models. And even when there are animal models, frequently the data that is gathered in preclinical trials does not correlate over to the human side. So you may get really good results in your animal trials, but when you go over to humans, you have less efficacy, or sometimes even no efficacy at all. Because our technology uses human-derived iPS cells as a starting material, we believe that we can use our platform to obtain human-relevant preclinical data and reduce the need for animal models. So without further ado, I would like to introduce our technology to you. We have two core technologies at Jiksak. The first is our nerve organoid technology, and these are in vivo-like nerves on a chip. Our second technology is human synapse beads, which is a human nerve synapse evaluation system. And I will talk about this later on in my speech today, but just to let you know, we do have this synapse evaluation system, and I'll talk a little bit about it during my nerve organoid speech. So to go into more detail about our nerve organoid technology, as I said, they're in vivo-like nerves on a chip, and that chip is a micro device that consists of 3 interconnected chambers. On the left is the well that we would deposit a iPS-derived nerve sphere into, and then as we culture this chip, we're going to have the axons start to extend out from the neural sphere, and they're going to extend down this microchannel. As they're extending down the microchannel, they are self-bundling, and then they're going to terminate into the well here on the right, the axon termini well, where they are growth cones. The growth cones terminate. We can do experiments right here in the chip on these nerve organoids, but our organoids are also robust enough to be removed from the device. As you can see in this microscopy picture, they can be removed intact. The cell body and axon bundle are still connected. Now, I would like to show you some time-lapse videos of our nerve organoids. Here we see the nerve sphere is actually not pictured here. It's cut off in this picture, but what we see here is the time-lapse video of the axons extending out from the cell body down through the microchannel, and they are cell autonomously self-bundling into an in vivo-like fascicle. So I'll let you look at that one more time. And then I'll move on to the next video. So As I mentioned, we can remove the organoid from the chip. It's going to still be intact. It's robust. You can manipulate it. And that's exactly what this is a video of, us taking an intact removed organoid from the chip. We're extending the axon back out to its full length here. And just for your reference, the axon bundle here is actually about the width of a human hair, about the same width. So what's so special about axon bundles anyway? Well, first of all, the name of our company, Jiksak, that means axon in Japanese. And with our technology, you can have a clear separation of the cell side and the axons, so the somatodendrites and the axon bundles. Not only that, but our technology does allow for recovery of enough axonal tissue to perform omics analysis. So I've got some pictures here of Western blot, so protein analysis to see the difference in protein expression on the cell side versus axons. And we can also look at RNA sequencing data of both the somatodendrites and the axons. Not only that, but you can perform standard pathophysiological and anatomical analyses on our organoids. And because they are not 2D, they are actually 3D structures, we can see very in vivo-like cell interactions when we perform co-cultures with target cell types. Okay, so now I would like to present two papers that have been recently published using our nerve organoid technology. The first paper, published in 2019, the researchers actually did a comparison between how much RNA could be retrieved from our nerve organoid versus in two other chips that were in the literature. And they found that starting with 1 × 10⁴ cells, nerve cells in our chip, they were able to retrieve 12 nanograms of RNA. Looking over here at this data for the chip here in the middle, they found that the literature stated that starting with 5 × 10⁵ cells, they could get 10 picograms of RNA. And finally, over here in this last one, last chip that they looked at, that has a relatively high yield of 80 to 100 nanograms of RNA, but the starting material is 2 × 10⁶ cells. So we took this to mean that our chips are able to provide a relatively high yield of RNA from a relatively low amount of starting cells. Now, to go into like the scientific research that was actually conducted in this paper. The researchers generated FUS-ALS motor neurons and grew them up into nerve organoids in our chips. They then separated the somatodendrites and the axons separately to perform RNA sequencing on somatodendrites and axons. Now, interestingly, they actually did find that there was a difference in the RNA transcript enrichment of somatodendrites versus axons. In particular, they found that extracellular matrix binding was particularly enriched in the somatodendrites, while neuropeptide hormone activity was enriched on the axons. So taking this data, they actually went in and looked for things that were specifically changed in the FUS mutant axons. And they focused on the transcripts that were both upregulated and which increased the gain of toxic functions in the axons. From this, they were able to identify that the FOS family of genes was of particular interest, and then went on to do further investigations, both in vitro and in vivo, which identified FOS-B as a key regulator in aberrant axon branching of FUS-ALS. So they felt that the conclusion here is it may be interesting to look at the FOS-B as a potential target for ALS therapies. In the second study here, which was published in May of this year, the researchers took patient-derived TRDBP mutant ALS cells, turned them to iPS cells, generated motor neurons, and then again grew them in our neuroorganoid device, again for the purpose of identifying separating the soma and the axons for RNA sequence. And from this, they found 61 candidate genes that they believe were involved in TRDP mutant axonal pathology. From these, they focused on FOX2B due to its reported role in neural crest and motor neuron differentiation, as well as the fact that FOX2B mRNA has a TDP43 binding sequence. Now, in the last talk, she was talking about the TDP43 and its role in not only ALS, but other neurodegenerative disorders. So we think this is very interesting data indeed. From this, the researchers concluded that downregulation of FOX2B results in shortened neurites, as well as a decrease in the function of motor neurons. And they might be of interest to look at the FOX2B pathway for ALS treatment. Now I would like to give a little bit more detail about our products. At the present time, we have 2 chips for sale. The first chip that I will talk about is our A chip. A chip have a long microchannel. It's 2 centimeters in length, and we can grow very long axons here. This is the chip that you would be doing the omics analysis if you were interested in that. We also have 2 live organoids that can be sold with this chip. The first is normal motor neurons, iPSC-derived motor neurons, and the second is patient-derived ALS motor neurons. So I'd like to show a little bit of data here. So with the normal motor neurons and the ALS motor neurons, they both grow fully in our chip without any problems. They grow with no statistically significant difference in the length of the organoid, nor in the width of the axon fascicle that is formed. So now, before I show you the next data, let me remind you, our second core technology is a system to analyze synapse formation in humans. So I haven't introduced that core technology yet. I'll introduce it in more detail in a minute, but we do have this technology, and we use that to actually look at the function of the ALS neurons and against regular motor neurons. And what we found was that while normal motor neurons can form very robust synapses, actually ALS motor neurons are unable to form as many synapses. Their synapse formation capacity is severely decreased compared to normal. Not only that, but when we look at the strength of the synapse formed, we see that even if they can form synapses, the synapses formed are weaker than for normal neurons. So we think that taken together, this means that our technology can be used to generate organoids which recapitulate the ALS phenotype. Next, I'll move on to our second chip, which we have for sale, the B chip. B chip have a much shorter microchannel than A chip, and thus cannot be used for omics analysis. However, the unique characteristic of our B chip is that there are 2 medium compartments which are completely separate, except through the microchannel. And so these are ideal for co-culturing cells. Let's say that you're interested in looking at the neuromuscular junction. So you would plate motor neurons over here, and you want to grow them, of course, in neuron maintenance medium. And then you're going to plate muscle cells over here on the axon side. And when you have those cells, you don't want to be growing them in neuron medium, you want to be growing them in muscle medium. So you can actually do that, and both cell types will be growing very happily, and you can do experiments to look at the neuromuscular junction. Furthermore, our B chips are designed to fit into a standard 96-well plate format, so you can perform standard imaging very easily with these chips. So the next data I'm going to show you is preliminary data of another cell type. So I'd like to briefly explain that before I move on to the data. In addition to motor neurons, we also have nociceptor sensory neurons, which we can grow up in our B chips into the 3D nerve organoids. Now, when researchers are looking at the sensory nerves, a lot of times they grow the cells in 2D culture, and then they perform stimulation experiments on them with bath applications. So they just add compounds directly to the 2D culture. So that means that the compounds are coming in direct contact with both the cell body and the axons. But in real life, in the human body, if you are having an itch sensation or an itch stimulation on your hand, for example, that stimulation is happening from the axon side. The axons are sending the signal back up to the cell body. So we questioned, is it possible for us to stimulate just on the axon side? We're going to add the compounds only over here to the axons. Will we be able to see any, firing of the cell body? And we found that, yes, indeed, we can see firing of the cell bodies after stimulating just the axon side. So what you're looking at here is calcium imaging of the same organoid being stimulated by two different stimulation compounds. So menthol here and histamine here. And we are seeing the firing of these axons, of the cell bodies. And another thing that we also found very interesting was what the parts of the cell body that we see firing in response to menthol are different than what is firing when we stimulate with histamine. So we believe that this really mimics the actual firing of sensory nerves in vivo. And so we hope to get this out for pain research, among others, in the near future. Now I'd like to talk about the last application that I will talk about today for our nerve organoids, which is how to apply axons, the axon portion, to peripheral nerve repair. And we're very excited about this application because this project was very recently accepted into a regenerative medicine accelerator program here in Tokyo, and we have some very promising preliminary data. So we grow out our nerve cells in our chips and we harvest the axon portion only. We embed that in a biocompatible matrix, and then we're going to implant it over to bridge the gap in a damaged nerve. And this product then acts as a scaffold for axonal regrowth. We believe that with this product, we will be able to bridge larger nerve gaps than what we're able to do currently with traditional nerve bridges. So one thing that we have very recently started investigating is the use of Elixirgen neurons in place of our current vendor for peripheral nerve repair. And what we found was our vendor A, current vendor, their nerves take about 3 to 4 weeks to grow up to the optimal length for use in the peripheral nerve project. However, when we looked at Elixirgen's nerves, we found neurons, we found that they grow to the optimal length in only 14 days. Not only that, but the width of the axon fascicle that is grown with Elixirgen cells is significantly thicker than what is formed with our current vendors, as seen in these enlarged photos. So since vendor A's cells are not ready to be used for 3 weeks, we decided to go ahead and compare side by side Elixirgen vendor A for 3 weeks. And we did find that Elixirgen significantly outperformed our current vendor in both the length and the width of the axon fascicle that was formed. Our next step is to investigate Elixirgen's use in the peripheral nerve repair project. So that finally brings me back to talk about our second core technology, our engineered human synaptogenic beads, which, as I very briefly talked about, they are a human nerve synapse evaluation system. So Jiksak's co-CEO, Dr. Norihiro Yumoto, who will be joining me today for the Q&A session, he did his postdoctoral research fellowship at New York University. And at that time, he was studying the neuromuscular junction, and he found that in mice, the LRP protein is a critical retrograde signal for presynapse formation on the neuromuscular junction. After he came back to Japan, he identified the human analog of mouse LRP4, and we call that protein X, and that is what we use to coat our synaptogenic beads. So a typical way that we would use the synaptogenic beads is to culture them with human motor or glutamatergic neurons that have the axons already grown out for them, so in 2D culture. And what we have found is that anywhere that the axons contact our protein-coated beads, the presynaptic machinery is formed. So I'd like you to look over here at the SEM data. So I'm talking about the control beads here. What I mean is we have coated our beads with a control protein that is not known to have any activity at the neuromuscular junction. Our active beads are the beads that have been coated with our protein X. And as you see, with the control beads, there's no synapse signal here. So there's no synapses forming between the axons and the beads. However, there's robust synapse formation as indicated by synapsin. This purple is the beads signal, and TUJ1 is axon signal. And when we merge that all together, we can see this yellow-white signal that is the synapse formation on the beads themselves. As I mentioned at the beginning of my speech today, our mission here at Jiksak is to look for therapies for ALS, to find cure even for ALS. So all of our in-house drug discovery capabilities are focused on therapies for ALS, finding compounds that can be used for ALS. So we used our synaptogenic beads to screen the FDA small molecule library for compounds that positively impact synaptogenesis. And we found 7 compounds that promote and 1 compound that suppresses synapse formation. We then went on to perform further testing in one of the compounds, thiamine hydrochloride. And we found that indeed, thiamine hydrochloride had a dose-dependent positive effect on synaptogenesis with the ALS motor neurons. So we believe that this data shows that actually our synaptogenic beads can be used for compound screening, and our hit compounds might be useful for not only ALS, but other synaptopathies, as well as, for example, age-related sarcopenia, which we believe acts on a similar mechanism of action. Now, the last thing I will cover today in my speech is an application for our synaptogenic beads in the early detection of chemotherapy-induced peripheral neuropathy, or CIPN. So when researchers are looking at compounds, chemotherapeutic compounds, to see if they're going to induce CIPN, the traditional way of doing that is to plate out the neurons in 2D, grow out the axons, add the chemotherapeutic agents, and then look for axon degradation or the reduction in axonal area. However, once the axons have already been degraded, it is considered very difficult to reverse that and to fix CIPN. So we wondered if there was a way that we could use our synaptogenic beads to screen for early stage of CIPN. So in this experiment, we took the nerve cells, we grew them out in 2D, grew out the axons, and then we added low doses of 3 chemotherapeutic compounds to the culture for 72 hours. As a positive control, we have our protein X-coated active beads with no compounds added. And as a negative control, we have the inactive protein-coated beads, the protein that has no activity at the neuromuscular junction. And we added no compounds to this culture either. So just looking at these pictures, it's quite difficult to tell, like, if there's any axon degradation happening here, is there any reduction in the axon area? And when we actually quantified that, we did indeed see that there was no statistically significant difference or reduction in the axon area for any of the compounds that were added. However, since we're looking at the synapse formation via our synaptogenic beads, we looked at that data and we found that actually, yes, we could detect that for compound B, there was a significant reduction in the number of synapses formed. That's very exciting for us. But you may be wondering, why is that exciting that you guys tested 3 compounds and only 1 compound reduced synapse formation? So actually with compound A is a chemotherapeutic agent, but it is not known to cause CIPN. So when we saw this, okay, this means our system is working here. But we did test 2 compounds that are known, that are associated with CIPN. And what we think is happening here is compound B is believed to act through the axon. It is believed that it causes CIPN, it damages the nerve through the axon. So this is very great data. Yes, it's acting through the axon. So we are seeing a reduction in synapse formed. Compound C, on the other hand, the mechanism of action is actually believed to be through the nerve itself. And that is how it's believed to kill the nerves from the cell body side, I should say. So we do think that this is actually great data, because we feel that our system can be used to screen for CIPN caused from the axon side, which makes sense since it's synapse formation. And we think that this has applications in both understanding the mechanisms behind CIPN, as well as potentially screening for compounds that can block CIPN, or perhaps even reverse it in the early stages. And I will just show you this data here, which will show you a little bit more about what I mean by synapse strength. So if you look here, this is the results of the screening of the 3 compounds with our synaptogenic beads. The positive control, all of these white, at the ends of all of these white arrows are the overlay of the synapse and beads. So we can see the white, yellowish white signal here. And we see quite a lot of the synapses formed here, as well as with compound A and compound C. We see lots of the synapses formed, and the signal at the end is quite intense and bright. However, looking at compound B, not only are there fewer synapses formed, but if you look closely, you can see that many of the synapses formed have a very weak signal when compared to the other compounds and positive control. And that is what we mean when we're talking about the strength of the synapses formed. Thank you so much for your kind attention during my speech tonight. At this time, I would actually like to introduce our co-CEO, Dr. Norihiro Yumoto, because he'll be joining me to answer your questions today. Dr. Yumoto earned his Ph.D. in medicine at Kyoto University in 2008. During his postdoctoral fellowship at New York University, he became interested in understanding the molecular mechanisms of neuromuscular synapse formation and maintenance. In particular, how motor nerve terminals recognize skeletal muscle and stay attached to them throughout life. Dr. Yumoto joined Jiksak in 2018 as Chief Technology Officer and transitioned to the co-CEO role in 2021. As part of his responsibilities at Jiksak, he studies molecules and signaling pathways that increase the connectivity between motor nerve terminals and skeletal muscles for first-in-class drug development for neuromuscular diseases. And now I will turn it back over to you, Michal, for the Q&A session. Thank you.
Hi, thank you so much. That was very interesting. Okay, we do have some questions coming in. First, just a general question about your business model. Do you offer products and services, or just products, or what is available?
We do offer both products and services. We are located in Japan. So at the moment, we do not have our live organoids to ship to the United States, but we are working on getting that up and running. We do have a distributorship agreement with Nakalai USA. And so they are able to distribute our empty chips. And then please feel free to contact us and we can work out our services. We can introduce our services to you as well.
Okay, great. So on slide 8, do you offer axonal trafficking assays?
Slide 8, let me go back to slide 8. Axonal trafficking assays. Dr. Yumoto, do you want to cover this?
Well, that we have not done it yet with our hands. However, I think it definitely feasible. If you are very interested in that particular asset, we can discuss and we can do some feasible study for future collaboration research.
Excellent. All right, thank you. On slide 11 and 12, what is the mutation in the ALS organoid?
Let's see, 11. Oh, yeah, here. This is actually sporadic ALS, so we do not know the actual mutation here. These are sporadic ALS patient.
Okay, thank you. On slide 28, is that assay able to be performed with live cell imaging or like using GFP tagged neurons?
We were actually just talking about this today, Dr. Yumoto and myself. So actually, Dr. Yumoto, you are the specialist here, so I will let you cover this one.
This one question is this a DSP tagged? This is the fixed, we analyze the synapse formation with the fixed neurons. However, probably you can use a lentivirus that let the cells express GFP tagged something synaptic protein that will allow you to visualize the live imaging. So again, it is definitely feasible, but we haven't done it with our hands.
Thank you. Here's another question. You evaluate the synapse strength in terms of the signal strength, but have you checked for correlations with the functional strength?
By functional strength, I imagine they mean maybe like uptake. And yes, we have done experiments with uptake, and we do know that the axons are functional in that respect. However, we've not done, as far as I'm aware, we've not actually looked at the correlation between the intensity of the signal in the immunohistochemistry and the functional uptake. Is that correct, Dr. Yumoto? We haven't done that, correct?
Yes. Yes.
That's an interesting idea, though, and definitely something we will look into.
Okay, thank you so much. That was very informative. And I wanted to just thank everyone for joining us today and remind you that this webinar and all of our webinars are available for viewing on our website and on YouTube. We hope you enjoyed it and learned a lot, and you can feel free to share this with any colleagues who may not have been able to join. Thank you so much. We wish you a wonderful weekend.
Thank you.
Thank you.
Thank you so much.